I'd say it's definitely very much an honor and privilege to, have the support to be able to work on these problems. And my name is Omar Abuddayeh. I'm a co-director of the Gutenberg Lab. Jonathan comes from a much more hardcore computational mathematical background. I come much more from this physical engineering background. When we're thinking through a problem, we'll have very different ideas to debug that problem.
In grad school, we've kind of had overlap for years. I would say, we had some cross-pollination with groping even before we moved to McGovern. A big other piece is that we had work on Rett's as a collaboration with Gail Mandel. We had a very personal experience. On a flight, we were seated next to a woman and her child flying to Boston for Rett's treatment. That was actually just a completely serendipitous, connection.
Rett syndrome, right, is a disorder that can be traced to genetic mutation. As genetic engineers, we've long been interested in, can we build genetic tools to permanently correct those mutations? Although it's there's a specific gene you can track this disease towards, there's actually many different mutations in the gene. One of the ways to address this challenge is to say, hey, instead of treating the actual mutation, can we just replace parts or even the entire gene at once with a single drug? And that would actually apply to all patients of the disease. And so our lab for many years has worked on these gene replacement strategies. It's taking gene editing to the next level. Maybe we can apply some of the stuff we learned from CRISPR and genome editing engineering to that space. And that's exactly what we did. We started seeing the results that made it seem like you could get therapeutically relevant levels of editing with this technology for things like Rett or Huntington's. Transplicing is one way that we actually do this. Splicing in itself is a natural phenomenon in the cells. And so a lot of the engineering is actually figuring out, okay, how can we interfere with the natural splicing process to make this replacement much higher efficiency? Now we're spending a lot of time trying to get it to work well with the MECP2 gene, which is the gene that drives Rett syndrome.
The way that the mutations are spread out in the MECP2 gene means that we can replace just one part of it and get a large number of mutations replaced in kind of one approach. We have ways that we can deliver these machinery to the brain very effectively. I think altogether makes it a very appealing target, MECP2, and creates a lot of potential, we hope, for Rett's patients.
Yeah, I mean, the ultimate goal in the short term is to really develop proof of concept data that our transplicing technology can actually get therapeutic levels of editing correction in the MECP2 gene. And then the long-term goal is, can we use that proof of concept data to actually fund efforts to develop this as a drug for Rett patients? So we need to show that it's first possible to even do this.
Beyond being kind of a fellow human and a fellow parent, as a scientist, there is some cautious optimism around how we can start to think about genetic diseases. Nothing is solved till it's solved. And even though we and many, many others in the community are working on approaches, we have to see them through. But there is hope there. And I think hope can be very valuable to people. We're very appreciative to the RSRT for support.